All right, welcome back, everyone. So we're going to begin, or continue rather, talking about blood and focusing here a little bit on some of the various parts of blood. So again, looking at maybe some plasma, some things in the plasma, and then we'll talk briefly about how we make some of our different types of blood cells, right? So recall plasma itself. Plasma is the aqueous, or the majority of the aqueous portion of blood, right? So it's primarily water, right? Primarily water. So this is the reason, recall, when we did our endocrine lecture, we talked about the fact that certain types of hormones, right? So we know that there are lipid-soluble hormones and water-soluble hormones. Water-soluble hormones were able to dissolve in the blood because it's primarily water, right? It's 92, 93% water. The remaining kind of 7% or so, or I guess remaining 8%, are plasma proteins and then other molecules and things dissolved in the blood. So proteins include things like antibodies, albumin, and then you have the various molecules of things like sodium, potassium, glucose, right? All these other things, amino acids, fatty acids that you have dissolved in the blood itself, right? And again, we're keeping those at a relatively constant level, right, or consistent level. Blood or the plasma itself is considered an extracellular fluid, right? So an ECF. So remember, there's extracellular fluid and there's intracellular fluid. And what this means is that extracellular fluid, it exists outside the cell, right? And that should make sense, right? So you have your red blood cells, you have white blood cells, you have platelets, and then you have your tissue cells, right, that make up your skin and muscles and whatnot. And this plasma is outside of all of that, right? Outside of all those cells. So it's extracellular fluid, right? And we know that there are other types of extracellular fluid, right? So there's interstitial fluid, there's cerebrospinal fluid, right? There's the fluid in the eye, right? The aqueous humor. All of that is fluid that exists outside the cell, right? Synovial fluid, all of that's outside the cell. So it's extracellular, right? And it's similar in composition to interstitial fluid, which is the fluid in between tissues. But importantly, plasma has a much higher protein concentration, and we'll look at some of those proteins coming up, okay? Blood as a whole, because of some of these proteins, is considered a colloid, right? So colloid is a type of mixture, if you will, that contains these various proteins, right? And so the presence of these proteins and the amount of the protein is going to give blood a what's called a colloid osmotic pressure. Sometimes it's just called an osmotic pressure, right? And so they're going to exert this pressure that's going to act to try to hold on to fluid in the blood, in the plasma. And we'll see that in just a little bit. And we'll talk about it again whenever we do blood vessels and we talk about capillary filtration. Other plasma proteins, we have albumins, so a major type of protein that's produced in the liver. We have globulins, so immunoglobulins are an example, so these are our antibodies. Fibrinogen, okay, we'll talk a lot about fibrinogen coming up whenever we do blood clotting. And then clotting proteins themselves. Which are produced in the liver, right? Along with many of these other types of proteins. So you can imagine that if many of these are produced in the liver, and you have some type of liver dysfunction, you have fatty liver disease, you have cirrhosis, cancer of the liver, something like that, then the composition of the blood and the function of those proteins is going to be affected. Right? And that should make pretty intuitive sense, right? Now, I mentioned just previously that the proteins in the blood, those plasma proteins, or some of them, help the blood exert what's called a colloid osmotic pressure, or just an osmotic pressure. And in essence, what this does is it is the pressure in the blood that helps to retain or hold on to fluid, right? So it prevents loss of fluid as blood moves throughout the capillaries, right? So on different sides of the capillary bed, as we'll see, there are different pressures for this colloid osmotic pressure. It's higher on one end than it is the other, and we'll look at that. But what this does, it helps maintain blood volume. So in other words, keeping the appropriate amount of fluid in the blood vessel, which then in turn assists with blood pressure, right? So blood pressure is really primarily dependent on, or largely dependent on, the amount of fluid in the blood, right? So in other words, the blood volume, right? So this osmotic pressure can be affected, as we said, if you have some type of liver disease that's affecting the production of the plasma proteins, or if you have an issue that has to do with the getting rid of the plasma proteins. So like a kidney disease where you filter out the plasma proteins, right? Now we'll see this whenever we do our renal lecture, our urinary system lecture, is that typically we don't have protein in the urine, right? Protein in the urine is referred to as proteinuria, and we typically don't have that, right? And that's a good thing because we don't want to get rid of these plasma proteins. But if you have some type of renal dysfunction, right, glomerulonephritis or some other type of maybe acute kidney injury, AKI, then you can have some protein end up in the urine. Now, if you don't have enough, or you don't have an adequate pressure because of these plasma proteins in the plasma, you can develop something called ascites, right? And so ascites is when you have this movement of fluid, or this accumulation of fluid outside the blood vessel, typically in the abdominal cavity, right? And so you see this again with patients who have liver disease, also due to poor nutrition as well. So if you don't consume adequate amounts of protein, you can develop something called ascites, right? So some examples of some of these plasma proteins include what are called albumins. This is the biggest subset of plasma proteins. These are largely produced in the liver, and these exert a very great colloid osmotic pressure. So again, helping to retain and hold on to some of those, or some of that fluid, right? These albumins, they're also, if you notice here, act as transport proteins for some lipids and hormones and ions. For the hormones, remember when we talked about lipid-soluble hormones? So think like testosterone, aldosterone, something like that, estrogen. Albumin was one of the carriers, right, that helped to transport that hormone in the bloodstream. You also have globulins. This is the second largest group of proteins in the blood, and many of these are antibodies, are immunoglobulins, which are part of our immune system. There are 5 different types of immunoglobulins: M, A, D, G, and E, sometimes abbreviated IG. So IgA, immunoglobulin A, IgG, immunoglobulin G. We'll talk about those later on. You also have what's called fibrinogen. Fibrinogen is a major protein that is necessary for our clotting process. Now notice here that the name of fibrinogen, the ending, it ends in ogen, O-G-E-N, okay, ogen. When you see that ending, O-G-E-N, that means that it is, almost think of it in its kind of inactive form, if you will, the inactive form. Fibrinogen specifically is soluble, meaning it dissolves in the blood, right? What we'll see whenever we talk about our clotting process is we take the soluble fibrinogen and we'll convert it into insoluble fibrin. So you just take off the ogen, you take off a sequence of amino acids, and fibrinogen becomes fibrin. It is now insoluble, it does not dissolve in the blood, and it helps to keep our clot intact. It helps to hold the platelets and the other cells in place, right? So that's a major role of fibrinogen. We will talk about that later on, right? And then you have some other miscellaneous proteins in the blood that we won't necessarily get into, right? Now, blood is a solution. We talked about it being a colloid earlier. It's also a solution. It contains all of these various different dissolved things in the blood, right? So these various molecules, so think like glucose and think fatty acids and amino acids. It contains the electrolytes, sodium, potassium, calcium, various other types of nutrients, fatty acids, gases, bicarbonate, carbon dioxide, oxygen, other waste products as well that we're transporting maybe to the spleen, or we're transporting it to the kidney, something like that, right? So there's a wide variety, a great variety of the things that we find in the blood. Okay, so consider some of these questions, and what we're going to continue here is looking at the process called hematopoiesis, right? So this prefix hemato, okay, the root word is kind of heme, and this is referring to blood, right? So hemato means blood. Poiesis means to produce, or to synthesize, or to create, or something like that, right? So this process of hematopoiesis means to make blood, right? And specifically, we're talking about making our different types of formed elements, right? So think our red blood cells, white blood cells, and our platelets, right? Now, I'm going to show you an image in the next slide, but essentially what this image is going to show is what is summarized here on this slide. The different lines, if you will, that give rise to our different types of blood cells, right? So they all start from what's called a hemocytoblast, right? Or this kind of common stem cell that is undifferentiated, meaning it's not one specific cell. It's not a red blood cell, it's not a white blood cell, it's not a platelet, but it can give rise to any of those, right? So you may see it called pluripotent. It can differentiate into different types. It's not destined to become just one, it's destined to give rise to all of them, right? And then there's what's called the myeloid line and the lymphoid line, giving rise to lymphocytes or everything else. Easy way to remember this, the lymphoid line gives rise to lymphocytes. Lymphoid, lymphocyte, you can see how we got there. The myeloid line is everything else. Okay, red blood cells, all of the other white blood cells other than lymphocytes, and then ultimately what will be our platelets, right? So here you can see this this kind of tree structure here, right? This kind of development and progression of our stem cell, right? So our stem cell is this cell right up top here, and then we have all of the cells that come from that, right? So again, here's our lymphoid line, giving rise to lymphocytes. Here's our myeloid line, giving rise to everything else, right? So platelets, red blood cells, and then all of the other white blood cells except lymphocytes, right? And then for lymphocytes, there are then different types of lymphocytes. There are T cells or T lymphocytes, B cells or B lymphocytes. There's natural killer cells. There's plasma cells. We'll talk about some of those later on. But for now, know all of our blood cells are ultimately derived from the same common stem cell, this hemocytoblast, right? Again, when you hear the term blast, think build, think essentially giving rise to, right? So hemo, again, blood, cyto, cell, and then blast is build. So this is this kind of blood cell building cell that gives rise to all of these other different types, right? Red blood cells, platelets, and then all of our white blood cells as well, right? So here we can kind of see a progression specifically for red blood cells. And when you talk about red blood cells or the development of red blood cells, you call that erythropoiesis, right? And that should make sense, right? Hematopoiesis, think blood. Erythropoiesis, we said that prefix erythro means red, right? So this is the development of our red blood cells, right? Now you can kind of see this progression. A few things that I want to point out. Okay, a few things that I want to point out. One is where we end. So I want to kind of start with the end here. We end with what's called the mature erythrocyte, so this down here. And notice it does not have a nucleus. Okay, it does not have a nucleus. All through this progression, okay, so through this myeloid stem cell, this proerythroblast, this basophilic erythroblast, polychromatic erythroblast, they all have a nucleus, right? When you get to this what's called an orthochromatic erythroblast, don't worry too much about the specific name, but when you get to this point, and this is what's called a reticulocyte, think of a reticulocyte as like an immature red blood cell, right? It's not fully formed yet. You lose the nucleus, right? Now think, why would you want to lose a nucleus? Okay, why would you start with a nucleus way back here and through all this time, and then why would you lose it, right? And so our red blood cell, they're the only mature cell in the body that lacks a nucleus, right? And you call a cell that lacks a nucleus, you call that cell anucleate or anucleated cell, right? A meaning without, right? So no nucleus. And it's the only cell that doesn't have a nucleus, right? So why, think physiology here, why would a red blood cell that lacks a nucleus, why might that be beneficial, right? Think of the function of red blood cells, right? And then we'll come back to that in just a little bit, right? So here's a little development, a kind of picture of the development of red blood cells. We make somewhere around 3 million red blood cells a second. We filter them through the spleen to get rid of them. We recycle parts of it, the iron and parts of the hemoglobin, but we're constantly making red blood cells. And we'll talk about some molecules, some hormones that can affect the process of erythropoiesis. Okay, so again, meaning the development of these red blood cells, right? Now, you also have this process called leukopoiesis, right? So if erythropoiesis was making red blood cells, leukopoiesis is probably making white blood cells, right? And so again, these are going to be our various types of white blood cells. So generally, our white blood cells, we have neutrophils, lymphocytes, monocytes, eosinophils, and basophils, right? And then you can differentiate those into what are called granulocytes and agranulocytes, depending on whether or not they have granules in their cytoplasm, right? Now, an easy way to remember which ones are granulocytes. So granulocytes are neutrophil, basophil, and eosinophil. Is they end in "phil" because they are filled with granules. Okay, they are called granulocytes because they are filled with granules, and they end in "phil," P-H-I-L. Right, so that's a little helpful way to remember. The ones that end in "cyte," so monocyte and lymphocyte, those are called agranulocytes. Again, the A meaning without, because they don't have granules in their cytoplasm. Right, and so we'll talk about that more later on when we talk about identifying different types of white blood cells, right? Again, for the lymphocytes, remember, this is the only cell that's derived from that lymphoid line that we looked at in the graph several slides ago. So those T lymphocytes, or I'm sorry, the lymphocytes themselves that are formed can then differentiate into what are called B cells or T cells. B lymphocytes or T lymphocytes. We will talk about these in much greater detail later whenever we get to our immune system lecture. We'll talk about types of B cells, we'll talk about types of T cells, again, which are all lymphocytes, and we'll talk about their specific function in the immune system. But we'll talk about those in a lot more detail in upcoming lectures, right? And then we have thrombopoiesis, right? So we have hematopoiesis as a whole, so making blood cells. We have erythropoiesis making red blood cells, we have leukopoiesis making white blood cells, and then we have thrombopoiesis making platelets, right, or our thrombocytes, right? Now, I mentioned earlier in a previous part, I think, that the platelets really aren't cells in the sense that they're derived from a cell, okay? So almost think of them as fragments, really. So they're all derived from what's called a megakaryoblast, or, which forms into a megakaryocyte. And then this megakaryocyte essentially increases in size. It increases in cytoplasm, and then it ultimately will fragment. It will, think of it almost as kind of exploding, for lack of a better word. It kind of bursts, and then all of these small little cytoplasmic fragments are what we have as thrombocytes, what we have as platelets. So that's why whenever you look at a blood smear, and you'll look at a blood smear in both lecture and lab, when you look at the blood smear, the platelets are tiny. They're incredibly small, much smaller than the red blood cells, which are typically smaller than white blood cells. But these platelets are incredibly small, little fragments of cells. So if you see those little kind of purple fragments floating in blood whenever you're looking at it as you're studying, those aren't just miscellaneous, random, non-important things. Those are the platelets, right? So pay attention to those whenever you see them, right? So again, here's a summary of all of those different cells that are being produced. Again, the lymphoid line you see on the right, the myeloid line you see on the left, giving rise to all the cells. Red blood cells, right? We have red blood cells here, we have platelets here, and we have all of our white blood cells here, lymphocytes, and then all the others, right? So eosinophils, basophils, neutrophils, and monocytes, right? Again, each coming down their own line. And then what we were just talking about with thrombopoiesis, right, with platelet formation, they are derived from these large megakaryocytes, right? Notice that they're these large kind of multi-lobed they have a large multi-lobe nucleus, right? And then they're going to begin to fragment off these little cytoplasmic extensions. They fragment, they form these really, really small fragments, and then that is what becomes the thrombocyte. Right? That's what becomes the actual platelet, right? So we're going to stop here, and then we will pick up in our next part talking in more detail about erythrocytes in particular.